Right now, at this very moment, trillions of cells in your body are orchestrating a symphony so complex that we've spent centuries trying to understand even its opening notes. Every breath you take, every thought that crosses your mind, every gentle flutter of your heart, all of it emerges from biological processes so intricate that they still humble our greatest scientists. Welcome to Sleep About Science. Tonight we're exploring 100 things that biology still can't fully explain, all mysteries that remind us just how much wonder remains in the natural world. And if you drift off to sleep while listening, that's perfectly wonderful. These mysteries will be here when you wake, still waiting to be solved. Before we begin, please make yourself comfortable wherever you are in the world. Adjust your pillows, pull up your blankets, and remember that this is educational content designed to accompany you gently into rest. If you haven't subscribed yet, you might want to, so you never miss these gentle journeys into the deepest questions of life itself. Now imagine for a moment that you're a detective, but instead of solving crimes, you're investigating the most profound puzzles of existence. Every living thing around you, from the smallest bacteria to the largest whale, from the quietest moss to your own magnificent brain, carries secrets that we're only beginning to glimpse. Let's start with something so fundamental that we take it completely for granted: consciousness itself. How does the soft grey matter inside your skull create the rich, vivid experience of being you? Scientists can map every neuron, measure every electrical impulse, catalogue every chemical reaction, and yet the hard problem of consciousness remains beautifully, stubbornly unsolved. We know that somehow, somewhere in the intricate dance of billions of neurons, subjective experience emerges, but how? The feeling of redness when you see a sunset, the sharp clarity of sudden understanding, the gentle ache of nostalgia, these inner experiences remain one of biology's most profound mysteries. But consciousness isn't a single, monolithic thing. It comes in layers like the soft blankets you might be nestled under right now. There's the basic awareness of sensations, the higher order thinking about your thoughts, the sense of self that persists through time, and the mysterious binding of all these separate processes into a unified experience. How does your brain take the scattered inputs from different senses and different brain regions and weave them into a single, coherent experience of being you? And consciousness isn't the only puzzle hiding in your brain. Consider sleep itself, this thing you might be drifting toward right now. Every night, you willingly surrender consciousness for hours, entering altered states that we're only beginning to understand. Why do we dream? Why do some dreams feel more real than waking life? Why do we forget most of our dreams, but remember others with startling clarity? Sleep seems to be essential for memory consolidation, immune function, and mental health, but the precise mechanisms remain wonderfully elusive. The architecture of sleep itself presents layers of mystery. We cycle through different stages of sleep throughout the night: night sleep, deep sleep, REM sleep, each with its own brainwave patterns and functions. But how does the brain orchestrate these transitions? What internal clocks and chemical signals coordinate the shift from wakefulness to sleep and back again? Why do we need different types of sleep? And what happens when we're deprived of specific stages? Then there's the question of how memories form and where they're stored. You can recall your childhood bedroom, the taste of your grandmother's cooking, the exact words of a song you haven't heard in years. But memories aren't stored like books on a shelf or files in a computer. They seem to be distributed across networks of neurons, somehow emerging from patterns of connection and electrical activity. How does your brain decide what to remember and what to forget? How do traumatic memories become so vivid they feel present, while other experiences fade like morning mist? The mystery deepens when we consider different types of memory. Does explicit memory mind the facts and events you can consciously recall, and implicit memory mind the skills and habits that guide your behavior without conscious awareness? How does the brain decide which memories become conscious and which remain hidden? Why can you forget someone's name but remember exactly how to ride a bicycle? How do memories change each time you recall them? And why do false memories feel just as real as true ones? Speaking of your brain, let's consider another mystery that might surprise you. We still don't fully understand how anesthesia works. Every day, millions of people undergo surgery peacefully unconscious thanks to anesthetic drugs that have been used for over a century. We know these drugs affect consciousness, but the precise mechanisms by which they turn off awareness while keeping vital functions running remain remarkably unclear. It's as if we've been using a master key to consciousness without fully understanding the lock. Different anesthetics seem to work through different pathways, yet they all achieve the same remarkable result. The temporary, reversible elimination of consciousness and pain sensation. Some anesthetics affect specific receptor channels, others seem to disrupt the normal patterns of brain activity, and still others appear to interfere with the brain's ability to integrate information. But how does any chemical substance reach into the deepest mysteries of consciousness and simply turn them off? Moving beyond the brain, let's explore the mystery of biological aging. Why do we age at all? From an evolutionary perspective, it seems disadvantageous. Wouldn't it be better if organisms could maintain themselves indefinitely? Some creatures, like certain jellyfish and turtles, seem to age so slowly they might be functionally immortal. Others, like mayflies, live gloriously brief lives. The cellular mechanisms of aging involve DNA damage, protein degradation, and cellular senescence. But why these processes evolved and why they proceed at different rates in different species remains an open question. The puzzle of aging becomes even more intriguing when we look at the cellular level. Your cells have built-in mechanisms for repair and maintenance. They can fix damaged DNA, replace worn-out proteins, and eliminate dangerous toxins. Yet these systems gradually become less efficient over time. Is aging simply the accumulation of damage that overwhelms repair systems, or is it a programmed process with its own evolutionary logic? Why do some cells seem to count their divisions and stop reproducing after a certain number? Consider the puzzle of biological clocks. Right now, even in the gentle darkness of your bedroom, your body knows what time it is. Every cell contains molecular clocks that tick away the hours, coordinating everything from hormone release to body temperature to immune function. But how did these clocks evolve? How do they stay synchronized? How do they adjust to different time zones or seasonal changes? The more we study circadian rhythms, the more intricate and mysterious they become. These biological clocks aren't just simple timers, they're sophisticated systems that can anticipate daily changes and prepare your body for what's coming. Your body temperature starts to rise before your normal wake time. Your digestive system prepares for meals at regular intervals, and your immune system follows its own daily rhythm. But how do these various clocks communicate with each other? How do they maintain coordination across all the different organs and systems in your body? And speaking of time, here's a biological mystery that touches every living thing. How do organisms know when to die? Cell death, or apoptosis, is a carefully programmed process. Cells literally commit suicide when they're damaged, infected, or simply no longer needed. This programmed cell death is essential for development menial fingers, formed because cells between them died at precisely the right moment. But how do cells know when their time has come? How do they execute this ultimate sacrifice so precisely? The machinery of cell death is remarkably sophisticated. Cells have multiple pathways for self-destruction, quality control mechanisms that assess when death is necessary, and signaling systems that can either promote or prevent cell death, depending on circumstances. But how did such a complex system evolve? Why would cells develop the ability to kill themselves? How do they avoid accidental activation of death pathways? Let's drift into the realm of development itself. Every human being begins as a single cell minus a fertilized egg, no bigger than the period at the end of this sentence. That one cell somehow knows how to become you, with your unique face, your particular way of thinking, your individual quirks and characteristics. The genetic instructions are there, written in DNA, but how does one cell know to become a brain cell while its neighbor becomes a heart cell? How do organs know when to stop growing? How does a developing embryo coordinate the formation of complex structures like eyes or hands or the intricate chambers of the heart? Development involves an intricate dance of cell division, migration, and differentiation. Cells must not only decide what type of cell to become, but also where to go and how to connect with their neighbors. How do migrating cells navigate through the developing embryo to reach their correct destinations? How do they know when they've arrived? How do different types of cells recognize each other and form the complex tissues and organs that make up your body? The mystery deepens when we consider how developmental programs are controlled. Genes don't simply turn on or off. They're regulated by complex networks of proteins and RNA molecules that respond to signals from other cells and from the environment. How do these regulatory networks evolve? How do they maintain precise control over development while allowing for the flexibility needed to respond to changing conditions? Even more mysteriously, how do some animals regenerate lost limbs while others cannot? A salamander can regrow an entire leg, complete with bones, muscles, nerves, and blood vessels. A starfish can regenerate from a single arm. Some worms can regenerate their entire head, including their brain. Yet mammals, despite our sophisticated biology, cannot regrow a lost finger. What evolutionary trade-offs led to such different capabilities? What molecular switches control regeneration, and might we someday learn to flip them? Regeneration isn't just about replacing lost parts, it's about recreating complex organized structures with all the right connections and proportions. How does a regenerating limb know how big to grow? How does it recreate the precise arrangement of bones, joints, and muscles? How do regenerated nerves find their way to the right targets? The more we study regeneration, the more it seems like a kind of biological magic. The mystery deepens when we consider metamorphosis. A caterpillar enters a chrysalis and emerges as a butterfly, not just different in appearance, but fundamentally reorganized at the cellular level. Most of the caterpillar's body actually dissolves during metamorphosis, broken down to raw materials that are rebuilt into something entirely new. How does this process maintain continuity of life while achieving complete transformation? How do the instructions for both caterpillar and butterfly coexist in the same genome? Metamorphosis involves some of the most dramatic transformations in biology. The caterpillar's crawling legs disappear, replaced by delicate wings. Its chewing mouthparts transform into a nectar-sipping proboscis. Its entire nervous system reorganizes to control flight instead of crawling. Yet somehow the butterfly retains memories from its caterpillar days. How is this continuity of experience maintained through such radical transformation? Speaking of transformation, let's explore the puzzle of cancer minus cells that have somehow forgotten the rules of normal growth and cooperation. Cancer cells divide when they shouldn't, ignore signals to stop, and invade tissues where they don't belong. But cancer isn't random chaos. It follows its own terrible logic, evolving and adapting like a separate organism within the body. Why do some people develop cancer while others don't? Why are some cancers so aggressive while others remain dormant for decades? How do cancer cells become so skilled at evading the immune system? Cancer presents a paradox of cellular behavior. The same mechanisms that allow normal development and tissue repair can go awry and create malignant growth. Cancer cells often reactivate developmental programs that should be shut off in adult tissues. They develop the ability to stimulate blood vessel growth, to resist cell death signals, and to invade and colonize distant tissues. How do normal cells acquire these abnormal capabilities? What sequence of changes transforms a healthy cell into a cancer cell? The evolution of cancer within the body follows its own terrible logic. Cancer cells with advantageous mutations outcompete their neighbors, leading to increasingly aggressive tumors. Some cancers develop resistance to multiple drugs simultaneously, while others split into genetically distinct subpopulations. How do cancer cells evolve so quickly? How do they coordinate their behavior to form complex tumor structures? Why do some cancers respond to treatment while others seem to anticipate and resist therapeutic interventions? This brings us to immunity itself, another beautiful biological mystery. Your immune system is an army of specialized cells that can recognize millions of potential threats, remember past encounters, and coordinate complex responses. But how does it distinguish between self and non-self? How does it know to attack a virus but leave your own cells alone? Autoimmune diseases occur when this recognition system goes awry, but we still don't fully understand what triggers these tragic cases of friendly fire. The immune system's ability to recognize danger is remarkably sophisticated. It can detect not just foreign invaders, but also signs of cellular stress, damage, or abnormal behavior. Immune cells patrol your body constantly, sampling proteins and other molecules to assess whether everything is normal. But how do they make these complex decisions? How do they distinguish between harmless foreign substances and dangerous threats? How do they avoid attacking beneficial bacteria while eliminating harmful ones? The mystery of immunity extends to allergies. Why do some immune systems treat harmless substances like pollen or peanuts as deadly threats? The hygiene hypothesis suggests that our modern sanitized environments might be confusing immune systems that evolved to fight constant threats, but this remains largely speculative. Why do allergies seem to be increasing in developed countries? Why do some children outgrow allergies while others don't? How do genetic factors interact with environmental exposures to determine who develops allergies? Autoimmune diseases present an even deeper mystery. In conditions like rheumatoid arthritis, multiple sclerosis, or type 1 diabetes, the immune system attacks the body's own tissues. What triggers this self-destructive behavior? Why do autoimmune diseases often target specific organs or tissues? How do genetic susceptibility and environmental triggers interact to cause autoimmune disease? Why do women develop autoimmune diseases more often than men? Let's journey deeper into the cellular realm. Every cell in your body contains mitochondria, tiny organelles that generate the energy that powers your life. These mitochondria have their own DNA, separate from the DNA in your cell nuclei, because they were once independent bacteria that entered into a partnership with early cells billions of years ago. But how did this partnership begin? How did two separate forms of life learn to cooperate so intimately? How do cells regulate the number of mitochondria they contain? And why do mitochondrial diseases affect some tissues more than others? The endosymbiotic origin of mitochondria represents one of the most important transitions in the history of life. Ancient bacteria somehow entered other cells, and instead of being digested or expelled, established a permanent partnership. Over time, most of their genes transferred to the cell nucleus, leaving them dependent on their host. But how did this gradual integration occur? How did the host cell learn to import proteins into mitochondria? How did the two organisms coordinate their reproduction so that daughter cells receive the right number of mitochondria? Mitochondria aren't just passive power plants. They're dynamic organelles that move around the cell, fuse with each other, and divide as needed. They have their own quality control systems and can even signal to the nucleus when they're damaged or stressed. How do mitochondria communicate with the rest of the cell? How do they coordinate their activities with cellular needs? Why do mitochondrial dysfunction and aging seem to be so closely linked? Even more fundamentally, we still don't fully understand how life began. The transition from chemistry to biology, from non-living molecules to self-replicating, evolving organisms, remains one of science's greatest mysteries. We know it happened at least once here on Earth, but how? Did life emerge in warm pools, deep ocean vents, or perhaps even in space? What was the first self-replicating molecule? How did the first cell membranes form? How did the genetic code evolve? The origin of life involves solving multiple puzzles simultaneously. Life requires metabolism to generate energy, heredity to pass information to offspring, and evolution to adapt to changing conditions. But how did these three essential features emerge together? Which came first, genes, proteins, or metabolism? How did the first cells maintain their integrity while allowing nutrients in and waste out? How did they avoid being poisoned by their own waste products? The mystery extends to the diversity of life itself. Why are there so many different species? Why did evolution explore so many different solutions to the challenges of survival? Consider the eye. Mitid has evolved independently dozens of times, creating compound eyes in insects, camera eyes in vertebrates, and mirror eyes in scallops. Yet each solution is beautifully adapted to its owner's needs. What drives this creative explosion of biological innovation? Evolution's creativity seems endless. There are fish that generate electricity, birds that navigate by magnetic fields, plants that trap and digest insects, fungi that glow in the dark, and bacteria that can survive in boiling water or pure acid. How did such extraordinary adaptations evolve? What environmental pressures shaped these remarkable solutions? How do organisms innovate while maintaining the basic functions necessary for survival? And speaking of innovation, how do new behaviors evolve? Birds build nests. Spiders spin webs, bees dance to communicate the location of flowers. These behaviors are often incredibly complex, requiring precise coordination and timing. But how did they first emerge? How are behavioral instructions encoded in genes? How do animals learn to modify inherited behaviors based on experience? The relationship between genes and behavior is far from simple. Most behaviors involve multiple genes working together, and the same genes often influence multiple behaviors. Environmental factors can turn genes on or off, and experience can modify both gene expression and neural development. How does this complex web of interactions produce the precise, adaptive behaviors we see in nature? Some behaviors seem to require cultural transmission, as well as genetic inheritance. Young animals often learn by watching their parents or other members of their social group. How do behavioral traditions arise and spread through populations? How do animals decide which behaviors to copy and which to ignore? How do cultural innovations interact with genetic evolution? Migration provides another set of puzzles. Arctic terns fly from Arctic to Antarctic and back each year, a journey of over 40,000 miles. Monarch butterflies navigate thousands of miles to wintering grounds they've never seen. Sea turtles return to the exact beaches where they were born. How do these animals navigate across vast distances? What internal compasses guide them? How do they account for weather, magnetic field variations, and other obstacles? The navigation abilities of migrating animals seem almost supernatural. They can maintain course across featureless oceans, compensate for wind drift, and arrive at specific destinations with remarkable precision. Some animals seem to use multiple navigation systems simultaneously: magnetic compasses, star maps, landmarks, and even infrasound or chemical cues. How do they integrate all this information? How do they calibrate their compasses? How do they deal with magnetic anomalies or cloudy skies that might interfere with their navigation? The mystery of animal navigation connects to broader questions about animal cognition. How do animals think? Do they have consciousness as we understand it? Elephants seem to mourn their dead, dolphins recognize themselves in mirrors, and crows use tools with remarkable sophistication. But how do we study the inner lives of other species? How do their minds work differently from ours? Animal cognition presents unique challenges for researchers. We can't ask animals about their thoughts or experiences, so we must infer their mental processes from their behavior. But how do we avoid projecting human experiences onto animals? How do we design experiments that are meaningful to animals rather than just to human researchers? How do we account for the different sensory worlds and communication systems of other species? Some animals display cognitive abilities that challenge our assumptions about intelligence. Clark's nutcrackers can remember the locations of thousands of cached seeds across vast territories. Honeybees can learn abstract concepts like same and different. African grey parrots can learn to use human language in apparently meaningful ways. How do these abilities develop? How do animal minds represent and manipulate information? Consider communication between species and even between different kingdoms of life. Plants can't speak, but they communicate constantly, Midas warning neighbors of insect attacks, sharing resources through underground fungal networks, and attracting specific pollinators with precisely tuned chemical signals. How did these communication systems evolve? How do plants know what signals to send and receive? Plant communication challenges our understanding of what communication really means. Plants release volatile chemicals that can warn neighboring plants of herbivore attacks, causing them to increase their defensive compounds before they're attacked themselves. How do plants decide whether to send these warning signals? How do they choose which chemicals to release? How do receiving plants interpret these signals and respond appropriately? The fungal networks that connect plants are mysteries themselves. These underground webs, sometimes called the Wood Wide Web, can span entire forests, connecting trees of different species in complex partnerships. Fungal trade nutrients with plants, receiving sugars in exchange for minerals and water. But how do these partnerships form? How do fungal and plants negotiate their exchanges? How do these networks process information and make decisions? Mycorrhizal networks display properties that seem almost intelligent. They can preferentially allocate resources to plants that need them most, broker exchanges between different plant species, and even seem to recognize kin relationships among plants. How do these networks coordinate their activities across vast distances? How do they store and process information? How do they make decisions about resource allocation? Even simpler organisms display behaviors that puzzle scientists. Bacteria can coordinate their behavior, forming biofilms and communicating through chemical signals. Slime molds, which aren't plants, animals, or fungi, can solve maze puzzles and optimize networks with no central nervous system at all. How do these organisms make decisions without brains? What does this tell us about the nature of intelligence itself? Bacterial communication, or quorum sensing, allows millions of individual bacteria to coordinate their behavior as if they were a single organism. They can time the release of toxins, coordinate the formation of biofilms, or collectively decide when to sporulate. How did these communication systems evolve? How do bacteria distinguish their own signals from those of other species? How do they prevent other organisms from eavesdropping on or jamming their communications? The puzzle deepens when we consider symbiosis-meiosis, the intimate partnerships between different species. Your own body hosts trillions of bacteria that help digest food, train your immune system, and even influence your mood. But how did these partnerships evolve? How do hosts and symbionts coordinate their needs? How do they maintain cooperation when evolutionary pressures might favor cheating? The human microbiome represents one of the most complex symbiotic relationships on Earth. Different bacterial species occupy different niches throughout your body, from your skin to your gut to your mouth. They help synthesize vitamins, break down complex carbohydrates, protect against pathogens, and regulate immune responses. But how is this community assembled? How do beneficial bacteria outcompete harmful ones? How does the microbiome change throughout your lifetime? Some symbioses are so intimate that it's hard to tell where one organism ends and another begins. Lichens are partnerships between fungi and algae so integrated that they were once thought to be single organisms. Corals are animals that depend entirely on photosynthetic algae living within their cells. How do such partnerships maintain stability across generations? How do partners coordinate their reproduction? How do they resolve conflicts of interest? The mystery of cooperation extends beyond symbiosis to social behavior. Why do some animals live in groups while others are solitary? How do social hierarchies form and change? Why do some animals sacrifice their own reproductive success to help relatives? Honeybee workers give up the chance to have offspring of their own to serve the hive, and naked mole rats live in colonies with social structures more like ant colonies than mammal societies. Social cooperation presents evolutionary puzzles. In a world where resources are limited and only the fittest survive, why would individuals ever help others? How do cooperative behaviors evolve and persist? How do social groups prevent freeloaders who take benefits without contributing? How do they maintain cooperation across generations? The evolution of eusociality medes give up their own reproduction to help relative cells represents one of the major transitions in the history of life. It has evolved independently in ants, bees, wasps, termites, and naked mole rats. What conditions favor the evolution of eusociality? How do individuals decide to become workers rather than reproductives? How do eusocial colonies coordinate the activities of thousands or millions of individuals? Speaking of reproduction, the diversity of reproductive strategies across life forms is staggering and often mysterious. Why do some species reproduce sexually, while others reproduce asexually? Why do some animals change sex during their lifetimes while others maintain the same sex throughout? Why did separate sexes evolve at all given the apparent efficiency of asexual reproduction? Sexual reproduction presents a fundamental puzzle. It seems incredibly wasteful, minus only half of your genes make it into each offspring, and you have to find a mate and court them successfully. Asexual reproduction would seem much more efficient, yet sexual reproduction is nearly universal among complex organisms. What advantages does sex provide that outweigh its obvious costs? How does sexual reproduction help organisms adapt to changing environments? The evolution of sexes themselves is mysterious. Why are there usually only two sexes rather than three or more? Why do some species have elaborate sex determination systems while others can change sex as needed? How do sex chromosomes evolve? Why do males and females often look so different when they share nearly identical genomes? Some organisms have evolved remarkable solutions to reproductive challenges. Seahorses have males that become pregnant and give birth. Some fish species consist entirely of females that reproduce by cloning themselves. Certain wasps can produce offspring without ever mating. How did these unusual reproductive strategies evolve? What advantages do they provide? How do they maintain genetic diversity? The timing of reproduction presents its own puzzles. Many animals have mating seasons precisely timed to environmental conditions, but how do they know when conditions are right? How do plants time flowering to maximize pollination while avoiding frost? How do animals coordinate breeding across entire populations? Reproductive timing often involves complex interactions between internal biological clocks and environmental cues. Animals may respond to changing day length, temperature, rainfall, or food availability. But how do they integrate all these signals? How do they predict future conditions based on current cues? How do they coordinate their timing with that of their mates and competitors? Parental care varies enormously across species, from parents that abandon their eggs immediately to parents that care for offspring for years. Emperor penguins incubate their eggs on their feet in Antarctic winter. Male seahorses carry developing young in specialized pouches, and some spider mothers allow their babies to eat them. What evolutionary pressures shaped these diverse strategies? How do animals decide how much care to provide? The evolution of parental care involves trade-offs between current and future reproduction. Caring for offspring takes time and energy that could be invested in producing more offspring. How do parents balance these competing demands? How do they assess the value of current offspring against future reproductive opportunities? How do they decide when to stop caring for one set of offspring and invest in the next? The mystery extends to how animals recognize their own offspring. In crowded penguin colonies, parents and chicks find each other through calls among thousands of nearly identical birds. How do they avoid raising the wrong chick? How do parents and offspring imprint on each other? What prevents parasitic species from exploiting parental care systems? Parent-offspring recognition often involves learning during critical periods early in development. But how do these learning mechanisms work? How do parents learn to recognize their offspring's unique signals? How do offspring learn to recognize their parents? What happens when these recognition systems fail or are manipulated by parasites? Let's explore another realm of mystery: the senses. We're familiar with the 5 human senses, but other animals perceive worlds we can barely imagine. Many animals see ultraviolet light, detect electrical fields, sense magnetic fields, or perceive infrasound. How did these exotic senses evolve? How do animals process such different sensory information? The diversity of sensory systems across life forms is staggering. Sharks can detect the tiny electrical fields generated by other animals' muscles. Platypuses use electroreception to hunt underwater with their eyes and ears closed. Migratory birds may see magnetic fields as patterns of light overlaid on their visual world. How do these sensory systems develop? How are they wired into the nervous system? How do animals learn to interpret such exotic sensory information? Echolocation in bats and dolphins creates detailed 3-dimensional maps of the environment using sound alone. How do these animals avoid interfering with each other's sonar? How do they process such complex acoustic information so quickly? How did echolocation evolve independently in such different animals? What does the world look like through echolocation? The computational challenges of echolocation are enormous. Bats must analyze the timing, frequency, and intensity of returning echoes to construct a detailed map of their environment. They must separate their own echoes from those of other bats, filter out irrelevant background noise, and do all this while flying at high speed through complex environments. How do their brains perform these calculations so quickly and accurately? The mystery of animal senses extends to pain and emotion. Do other animals feel pain as we do? Do they experience joy, fear, or sadness? How can we study subjective experiences in creatures that can't tell us about their inner lives? The question becomes more complex when we consider animals with very different nervous systems. Do insects feel pain? What about fish? What about plants, which respond to damage but have no nervous systems at all? Pain serves important functions in alerting organisms to damage and motivating protective behaviors. But pain also involves subjective experience that goes beyond simple reflexes. How can we distinguish between reflexive responses to harmful stimuli and genuine subjective pain? How do we design ethical experiments that respect animal welfare while advancing scientific understanding? The question of plant pain and sensation presents unique challenges. Plants respond to damage, competition, and environmental stress in sophisticated ways. They can communicate distress to neighboring plants and mount complex defensive responses. But without nervous systems, how do they process information and coordinate responses? Do they experience anything analogous to pain or stress? This brings us to the boundary between plant and animal behavior. Plants move, communicate, and respond to their environment in ways that can seem almost intelligent. They grow toward light, send roots toward water, and even seem to remember past experiences. But without nervous systems, how do they process information and make decisions? How do they coordinate complex behaviors across their entire structure? Plant behavior challenges our assumptions about what constitutes intelligence and decision-making. Plants can learn from experience, anticipate future conditions, and make choices that affect their survival and reproduction. But how do they do this without brains? What kinds of information processing systems have evolved in plants? How do they store and retrieve information? The Venus flytrap can count mind at N. It won't close its trap until 2 trigger hairs are touched within a certain time period. How does a plant without a brain keep track of stimuli over time? How does it decide when to act? Other plants can learn to ignore repeated non-threatening stimuli, suggesting forms of habituation and memory. How do these learning mechanisms work at the cellular level? Moving to the molecular level, we encounter even more mysteries. DNA carries the instructions for life, but how does it know when and where to express different genes? Gene regulation is incredibly complex, involving multiple layers of control that can turn genes on or off in response to environmental conditions. But how did this regulatory system evolve? How do cells maintain precise control over thousands of different genes? Gene regulation involves a complex interplay of proteins, RNA molecules, and DNA sequences that form intricate regulatory networks. Transcription factors can activate or repress gene expression, but how do they know which genes to target? How do regulatory networks evolve new functions while maintaining essential cellular processes? How do cells coordinate gene expression across different pathways and in response to multiple signals? Epigenetics adds another layer of mystery. Environmental factors can influence gene expression in ways that persist across generations. But how? How do cells remember environmental influences? How do these epigenetic marks get passed from parent to offspring? What role do epigenetic changes play in evolution and adaptation? Epigenetic mechanisms include DNA methylation, histone modifications, and regulatory RNAs that can alter gene expression without changing the DNA sequence itself. These modifications can be influenced by diet, stress, exposure to toxins, and many other environmental factors. But how do cells decide which genes to modify epigenetically? How are these modifications maintained through cell division? How do they get reset or transmitted across generations? The mystery deepens when we consider that most of our genome doesn't code for proteins at all. This junk DNA was once thought to be evolutionary debris, but it's increasingly clear that much of it has important functions we don't understand. How does non-coding DNA influence gene expression? What other roles might it play? Why is the genome so much larger than seems necessary to encode all our proteins? Non-coding DNA includes regulatory sequences that are transcribed into regulatory RNAs, and vast regions of repetitive sequences whose functions remain mysterious. Some non-coding sequences seem to act as spacers or structural elements. Others may serve as reservoirs for evolutionary innovation. How do we distinguish functional non-coding DNA from truly neutral sequences? How do regulatory sequences evolve and acquire new functions? Even the proteins that genes code for remain mysterious in many ways. How do long chains of amino acids fold into complex three-dimensional structures? How do they know what shape to take? How do proteins find their targets in the crowded environment of the cell? How do they work together in the intricate molecular machines that power life? Protein folding presents one of the most fundamental puzzles in biology. The sequence of amino acids determines the final structure, but predicting that structure from the sequence remains one of the greatest challenges in computational biology. How do proteins fold so quickly and accurately? What happens when they misfold? How do cells prevent and correct protein misfolding? The crowded environment inside cells presents additional challenges for protein function. Cells are packed with thousands of different proteins, nucleic acids, and other molecules, all moving and interacting in complex ways. How do proteins find their specific binding partners in this molecular chaos? How do they avoid inappropriate interactions? How do cells organize their internal space to facilitate proper protein function? The origin of genetic code itself is another puzzle. Why do 3 DNA letters code for each amino acid? Why are there exactly 20 standard amino acids? Why is the genetic code nearly universal across all life? These seem like arbitrary choices, but they must have been shaped by evolutionary forces we don't fully understand. The genetic code shows evidence of optimization. Unisimilar amino acids tend to have similar codons, so mutations are less likely to have severe effects. But how did this optimization arise? Was the current genetic code selected from many alternatives, or did it arise through historical accident and become frozen in place? How do we explain the few deviations from the universal code found in some organisms? Viruses present their own set of mysteries. Are they alive? They can't reproduce without hijacking cellular machinery, yet they evolve and adapt like living organisms. How did viruses originate? Are they simplified descendants of early cells, or are they escaped genetic elements? How do they evolve so quickly while maintaining essential functions? The relationship between viruses and their hosts is complex and often surprising. Many viruses remain dormant in cells for years before activating. What triggers their activation? How do cells detect and defend against viruses? Why do some viral infections become chronic while others are quickly cleared? How do viruses overcome host defenses while avoiding destroying their hosts too quickly? Some viruses seem to benefit their hosts rather than harming them. Viruses in the gut microbiome may help regulate bacterial populations. Some viral infections may provide protection against other pathogens. How do these beneficial relationships evolve? How do viruses balance their own reproduction with host welfare? Prions present an even stranger puzzle. These infectious proteins can cause devastating diseases like mad cow disease and Creutzfeldt-Jakob disease. But how can a protein be infectious? How do misfolded proteins convince normal proteins to change their shape? How did evolution produce organisms vulnerable to such an unusual form of infection? Prion diseases challenge our understanding of what constitutes an infectious agent. Unlike viruses or bacteria, prions contain no genetic material. They're simply proteins that have adopted an abnormal shape, yet they can replicate this abnormal shape by converting normal proteins. How do they cross species barriers? Why are some individuals more susceptible than others? How can we develop treatments for diseases caused by misfolded proteins? The immune system's ability to generate antibodies presents another molecular mystery. Your immune system can produce antibodies against threats it has never encountered before. It does this by generating enormous diversity through genetic recombination. But how does it create so much diversity without destroying essential genes? How does it avoid producing antibodies against your own tissues? Antibody diversity is generated through a remarkable process of controlled genetic chaos. Immune cells cut and paste their DNA in billions of different combinations, creating an astronomical number of different antibodies. But this process must be carefully controlled to avoid damaging the cells or creating autoimmune responses. How do immune cells balance diversity generation with genomic stability? How do they select the most useful antibodies from this vast repertoire? The development of immunological memory presents additional mysteries. How do immune cells remember past encounters with pathogens? How do they maintain these memories for decades? How do they distinguish between memory cells that should persist and those that can be discarded? How do memory cells know when to activate during subsequent infections? Wound healing showcases another complex biological process we don't fully understand. When you get a cut, multiple types of cells coordinate to stop bleeding, fight infection, and repair damaged tissue. But how do they know what to do? How do they communicate with each other? How does the healing process know when to stop? Wound healing involves a carefully orchestrated sequence of cellular behaviors. Platelets aggregate to stop bleeding, immune cells clear debris and fight infection, and various cell types migrate into the wound to rebuild damaged tissue. But how is this sequence coordinated? How do cells know when each phase is complete? How do they avoid overhealing or underhealing? The mystery extends to tissue regeneration and repair throughout life. Your skin completely replaces itself every few weeks, your gut lining renews every few days, and your red blood cells live only about 4 months. How do tissues maintain their structure and function while constantly replacing their components? How do they maintain the right number of each type of cell? Tissue homeostasis requires constant monitoring and adjustment. Stem cells must decide when to remain dormant and when to produce new cells. They must choose what type of cell to become based on local signals and tissue needs. But how do these decisions get made? How do tissues count their cells and maintain proper proportions? How do they respond to injury or disease while maintaining normal function? Cancer again appears as a breakdown of these normally precise control systems. How do normal cells transform into cancer cells? What early changes set them on the path to malignancy? Why do some organs develop cancer more often than others? How do cancer cells develop resistance to treatments? Cancer development typically involves the accumulation of multiple genetic and epigenetic changes over time. But how many changes are required? Which changes are most important? How do cancer cells acquire the ability to invade other tissues and metastasize to distant sites? Why are some cancers more aggressive than others? The heterogeneity of cancer presents additional challenges. Even within a single tumor, cancer cells can be genetically diverse, with different subpopulations having different properties. How does this diversity arise? How do different cancer cell populations interact with each other? How does tumor heterogeneity affect treatment responses? The mystery of individual variation adds another layer of complexity. Why are no two individuals identical, even identical twins? How does genetic diversity arise and persist in populations? How do populations adapt to changing environments? What determines which individuals survive and reproduce? Genetic diversity arises through mutation, recombination, and gene flow between populations. But how much diversity is optimal? Too little diversity makes populations vulnerable to disease or environmental change, but too much diversity might disrupt beneficial gene combinations. How do populations balance these competing pressures? How do they maintain diversity while preserving adaptive traits? The role of neutral evolution adds another layer to the mystery. Many genetic changes may have little effect on survival or reproduction, allowing them to drift randomly through populations. How much of evolution is driven by selection versus random drift? How do we distinguish between adaptive and neutral changes? What role does population size play in determining the relative importance of selection versus drift? Sexual selection presents its own puzzles. Why did elaborate courtship displays evolve? Why do males in many species compete for females rather than the reverse? Why do some species have extreme sexual dimorphism while others show little difference between sexes? How do preferences for certain traits evolve and spread through populations? The peacock's tail is a classic example. It's large, colorful, and seemingly maladaptive, yet it evolved because females prefer males with impressive tails. But why did this preference evolve? How do costly displays honestly signal male quality? How do such preferences persist despite their apparent costs? What prevents cheating or the evolution of less costly alternatives? Sexual selection can drive the evolution of traits that seem to contradict natural selection. Males may evolve elaborate ornaments, dangerous weapons, or risky behaviors that reduce their survival but increase their mating success. How do these opposing forces balance out? What determines whether sexual selection or natural selection is stronger in any given case? Cooperation and altruism present evolutionary puzzles. Why do individuals sometimes help others at their own expense? How did cooperation evolve in a world where selfish behaviors might seem more advantageous? How do animal societies maintain cooperation and prevent cheating? The evolution of cooperation requires overcoming the temptation to cheat, to take benefits without paying costs. Various mechanisms can promote cooperation, including kin selection, reciprocity, and group selection. But how do these mechanisms work in practice? How do organisms recognize relatives, remember past interactions, or coordinate group behaviors? How do they prevent exploitation by cheaters? The mystery extends to human behavior and psychology. How did our complex social behaviors evolve? Why do we have language, art, music, and religion? How do our brains create culture and pass it from generation to generation? What makes human cognition unique among animals? Human cultural evolution presents unique puzzles. Unlike genetic evolution, cultural traits can be transmitted horizontally between unrelated individuals and can change much more rapidly than genetic traits. How do cultural innovations arise and spread? How do they interact with genetic evolution? How do human societies maintain cultural stability while allowing for innovation and change? Language itself is deeply mysterious. How did the capacity for language evolve? How do children learn language so effortlessly? How does the brain represent and process linguistic information? Why do humans have such a powerful drive to communicate? What makes human language different from animal communication systems? The recursive nature of human language, our ability to embed ideas within ideas infinitely, seems unique among animal communication systems. How did this capacity evolve? What neural mechanisms support recursive thinking? How do children acquire this ability with no explicit instruction? Why did such a complex communication system evolve when simpler systems might seem adequate? The origin of creativity and artistic behavior puzzles biologists. Why do humans create art, music, and literature? Do other animals have aesthetic preferences? How do creative abilities develop? What evolutionary advantages might artistic behaviors provide? Human artistic behavior seems to violate simple evolutionary logic. Creating art takes time and energy that could be spent on survival and reproduction. Yet artistic behavior appears in all human cultures and seems to be intrinsically rewarding. How did these behaviors evolve? Do they serve hidden functions that we don't recognize? How do aesthetic preferences develop and change over time? Religious and spiritual experiences present another puzzle. Why do humans across all cultures develop religious beliefs and practices? What brain mechanisms underlie spiritual experiences? How do religious beliefs influence behavior and social organization? What role does religion play in human cooperation and group cohesion? Religious experiences often involve altered states of consciousness, feelings of transcendence, and encounters with the sacred. These experiences can be profoundly meaningful to individuals and can influence their behavior throughout their lives. But how do these experiences arise in the brain? What evolutionary functions might they serve? How do religious traditions maintain stability while adapting to changing circumstances? The mystery of human consciousness returns us full circle to where we began. What makes human consciousness special? How did self-awareness evolve? Why do we have subjective experiences at all? How do billions of neurons create a unified sense of self? Human consciousness seems to involve levels of awareness and self-reflection that may be unique among animals. We can think about our own thinking, imagine counterfactual scenarios, and contemplate abstract concepts. How did these higher-order cognitive abilities evolve? What neural mechanisms support them? How do they develop during childhood and adolescence? Dreams remain particularly mysterious aspects of consciousness. Why do we dream? Why are dreams often bizarre and illogical? How does the brain create the vivid experiences of dreams? Why do we forget most dreams but remember others clearly? What functions might dreaming serve for memory, learning, or emotional processing? The content of dreams often reflects our daily experiences, but transformed in strange and unexpected ways. People and places from different times and contexts get mixed together, impossible events seem perfectly normal, and emotional themes often dominate over logical narratives. How does the brain construct these dream experiences? What determines which memories get incorporated into dreams? Why are dream narratives often so different from waking thought? Memory formation and retrieval continue to puzzle neuroscientists. How are memories encoded in neural networks? How does the brain decide what to remember and what to forget? Why are some memories vivid and lasting while others fade quickly? How do false memories form? And why do they feel just as real as true memories? The formation of episodic memories, memories of specific events, involves the hippocampus and associated structures. But how do these brain regions bind together the different elements of an experience into a coherent memory? How do they tag memories with information about when and where they occurred? How do they link related memories together while keeping them distinct? Memory is not just about storing information, it's also about forgetting. The brain actively forgets information that is no longer relevant or useful. But how does it decide what to forget? How does forgetting work at the cellular level? Why do some traumatic memories seem impossible to forget while other important information slips away? The placebo effect demonstrates the mysterious connection between mind and body. How can belief alone influence physical symptoms? What brain mechanisms mediate placebo effects? Why do some people show stronger placebo responses than others? How can we harness placebo effects therapeutically while maintaining scientific rigor? Placebo effects can influence pain, immune function, hormone levels, and many other physiological processes. The effects seem to involve the brain's ability to modulate its own responses based on expectations and beliefs. But how do psychological factors translate into biological changes? How do expectation and belief get converted into neural signals that affect the body? The nocebo effect (MOT), where negative expectations lead to adverse outcomes, presents the flip side of this puzzle. How can negative beliefs cause real physical symptoms? What protects some people from nocebo effects, while making others vulnerable? How do cultural beliefs about illness and treatment influence placebo and nocebo responses? Phantom limb sensations highlight another puzzle about the brain's body representation. How does the brain map the body? Why do amputees sometimes feel sensations in missing limbs? How plastic is the brain's body representation? Can it be modified through training or experience? The brain maintains detailed maps of the body that can persist even after body parts are lost. These phantom sensations can include touch, temperature, position, and even pain in missing limbs. But how are these body maps maintained? Why don't they simply disappear when body parts are lost? How do they sometimes adapt to new body configurations? Mirror therapy and other treatments can sometimes reduce phantom limb pain by providing visual feedback that conflicts with phantom sensations. How does this visual information influence the brain's body map? What does this tell us about how different senses interact to create our sense of embodiment? The development of personality and individual differences presents ongoing mysteries. Why do people develop different personality traits? How much is determined by genes versus environment? How stable is personality across the lifespan? What brain mechanisms underlie personality differences? Personality seems to be influenced by both genetic factors and environmental experiences, but the interactions between genes and environment are complex and poorly understood. How do genetic predispositions interact with family environment, peer relationships, and cultural factors to shape personality? How do early experiences influence personality development? Can personality change throughout adulthood? Twin studies suggest that many personality traits are substantially heritable, but the specific genes involved remain largely unknown. How do multiple genes with small effects combine to influence personality? How do gene-environment interactions shape personality development? Why are some traits more heritable than others? Mental illness remains poorly understood despite decades of research. What causes depression, anxiety, schizophrenia, and other mental health conditions? Why do treatments work for some people but not others? How do genes and environment interact to influence mental health? What role do neurotransmitters, brain circuits, and developmental factors play in mental illness? The complexity of mental illness presents enormous challenges for researchers. Most mental health conditions seem to involve multiple genes, multiple brain systems, and complex interactions with environmental factors. How do we untangle these complex causal webs? How do we develop treatments that are effective for the diversity of individuals with mental health conditions? The stigma surrounding mental illness adds additional layers of complexity. How do social attitudes about mental illness affect diagnosis, treatment, and recovery? How do cultural factors influence the expression and experience of mental health conditions? How can we reduce stigma while advancing scientific understanding? The mystery extends to normal variations in human psychology. Why are some people more introverted, while others are extroverted? Why do people have different cognitive strengths and weaknesses? How do cultural factors influence psychological development? What determines individual differences in creativity, empathy, and resilience? Cognitive differences between individuals are substantial and consistent across time. Some people excel at spatial reasoning, while others are better at verbal tasks. Some are more analytical, while others are more intuitive. How do these differences arise? How much can they be modified through training or experience? What advantages do different cognitive styles provide? Cultural psychology reveals that many psychological processes that seem universal are actually shaped by cultural factors. How do cultural values and practices influence perception, cognition, and emotion? How do children acquire their culture's psychological patterns? How do psychological differences between cultures arise and persist? Love and attachment present their own biological puzzles. How do pair bonds form? What brain mechanisms underlie romantic love? Why do some relationships last while others don't? How did human mating strategies evolve? What role do hormones like oxytocin and vasopressin play in bonding? Human pair bonding seems to involve multiple neural systems that evolved for different purposes. The brain systems involved in romantic attraction overlap with but are distinct from those involved in long-term attachment. How do these different systems interact? How do they change over the course of a relationship? What determines whether initial attraction develops into lasting love? Cross-cultural studies reveal both universal and culturally variable patterns in love and relationships. What aspects of love and attachment are universal versus culturally constructed? How do arranged marriages and love marriages differ in their psychological dynamics? How do cultural values about relationships affect individual experiences of love? The parent-child bond showcases another mysterious aspect of human biology. How do parents and children develop strong emotional connections? What hormones and brain circuits mediate parental behavior? Why do some parents struggle with bonding while others bond easily? How do early attachment experiences influence later relationships? Parental behavior involves dramatic changes in brain structure and function, particularly in regions involved in caregiving and social cognition. But how do these changes occur? How do they differ between mothers and fathers? How do they vary across individuals and cultures? What happens when these bonding systems don't function properly? Attachment patterns established in early childhood seem to influence relationships throughout life. But how do these early experiences get encoded in the brain? How do they influence later behavior? How much can insecure attachment patterns be changed through later positive experiences? Human development from birth to adulthood involves countless mysteries. How do children develop theory of mind, the understanding that others have different thoughts and beliefs? How do moral reasoning and empathy develop? What drives the dramatic brain changes of adolescence? How do cognitive abilities change across the lifespan? The development of social cognition involves understanding that other people have minds with thoughts, beliefs, and desires that may differ from one's own. This understanding seems to emerge gradually during childhood. But how? What brain mechanisms support theory of mind? Why do some individuals with autism have difficulty with theory of mind while excelling in other cognitive areas? Moral development presents additional puzzles. Children seem to develop moral intuitions about fairness, harm, and cooperation from an early age. But how do these moral intuitions arise? How do they vary across cultures? How do they interact with reasoning and emotion to guide moral behavior? Adolescence involves dramatic changes in brain structure and function, particularly in regions involved in decision-making, risk assessment, and social cognition. But what drives these changes? Why does adolescence involve increased risk-taking and emotional intensity? How do these brain changes prepare adolescents for adult responsibilities? Aging brings its own set of biological mysteries. Why do some people age more successfully than others? What determines lifespan? How do genetics and lifestyle interact to influence aging? Why do some cognitive abilities decline with age, while others remain stable or even improve? Successful aging involves maintaining physical health, cognitive function, and social engagement throughout later life. But what factors promote successful aging? How do genes and environment interact throughout the lifespan to influence aging outcomes? What role do stress, diet, exercise, and social relationships play in healthy aging? Cognitive aging presents particularly intriguing patterns. Some abilities like processing speed and working memory tend to decline with age, while others like vocabulary and general knowledge often remain stable or improve. Why do different cognitive abilities show different aging patterns? How can we distinguish normal aging from pathological changes? What interventions might promote cognitive health in aging? The end of life presents final mysteries. What determines when someone dies? How does the body shut down during the dying process? What happens to consciousness as the brain stops functioning? These questions touch the deepest mysteries of biological existence and the boundary between life and death. The dying process involves the gradual failure of multiple organ systems, but the sequence and timing can vary greatly between individuals. What factors determine how the dying process unfolds? How does the brain maintain consciousness as other systems fail? What role do factors like social support, meaning-making, and spiritual beliefs play in the dying process? Near-death experiences reported by some individuals during cardiac arrest or other life-threatening situations raise additional questions about consciousness and dying. How do we study these subjective experiences scientifically? What brain mechanisms might underlie them? What do they tell us about the nature of consciousness and the dying process? Throughout all these mysteries runs a common thread: the incredible complexity and beauty of life itself. Every biological process, from the simplest chemical reaction to the most complex behavior, emerges from intricate networks of interactions that we're only beginning to understand. The mysteries of biology remind us that despite all our scientific progress, the natural world remains full of surprises. Each discovery opens new questions. Each answer reveals new layers of complexity. The more we learn about life, the more we realize how much we still don't know. Some mysteries may be solved in your lifetime through advances in technology, theory, and experimental methods. Others may take centuries to unravel as we develop new ways of studying living systems. Still others may always remain mysterious, reminding us of the limits of human understanding and the endless wonder of the natural world. The tools of modern biology are remarkably powerful. We can sequence entire genomes, watch individual molecules move inside cells, measure the activity of single neurons, and manipulate genes with precision. Yet even with these tools, many fundamental questions remain unanswered. How does complexity emerge from simplicity? How does life maintain itself in a universe trending toward disorder? How did consciousness arise from unconscious matter? As you drift toward sleep, surrounded by these beautiful mysteries, remember that your own body embodies countless unsolved puzzles. Your breathing, your heartbeat, your dreams, all of them involve processes that science is still working to understand. The bacteria in your gut are communicating in languages we're only beginning to decode. Your immune system is making decisions using criteria we don't fully comprehend. Your brain is processing information through mechanisms we're still trying to unravel. You are not just an observer of these mysteries. You are a living embodiment of them. Every cell in your body carries secrets. Every thought in your mind emerges from mechanisms we don't fully comprehend. Every day of your life you participate in the grand biological experiment that is existence itself. These mysteries aren't problems to be frustrated by minus. They're invitations to wonder. They remind us that despite all our knowledge, the universe remains full of surprises, full of beauty we haven't yet discovered, full of connections we haven't yet made. They humble us while simultaneously inspiring us to keep searching, keep questioning, keep marveling at the extraordinary complexity and beauty of life. The mysteries of biology connect us to the deepest questions about existence itself. How did life begin? How does consciousness arise? What is the nature of self? How do we balance individual needs with collective welfare? These aren't just scientific questions. They're philosophical and spiritual questions that have inspired human thought for millennia. Science provides one lens for exploring these mysteries, but it's not the only lens. Art, literature, music, and spiritual traditions offer their own ways of engaging with the fundamental questions of existence. The mysteries of biology invite us to use all our human capacities, rational and intuitive, analytical and creative, individual and collective, to explore the nature of life. Sleep well, knowing that tomorrow scientists around the world will wake up and continue working on these puzzles. Some will make small discoveries, others will have breakthrough insights. All will be participating in humanity's greatest adventure, the quest to understand life itself. Graduate students will stay up late, watching cells divide under microscopes. Field biologists will trek through rainforests cataloging new species. Computational biologists will develop new algorithms to analyze the vast datasets generated by modern experimental techniques. But science is not just the work of professional scientists. Every time you observe the natural world with curiosity and wonder, you're participating in the scientific enterprise. Every question you ask about how things work, every moment you spend marveling at the complexity of life, you're adding to humanity's collective engagement with these mysteries. The democratization of scientific tools and information means that more people than ever can participate in biological research. Citizen science projects allow ordinary people to contribute to scientific discoveries. Online databases make research findings accessible to anyone with an internet connection. Educational resources help people of all ages explore biological concepts and develop scientific thinking skills. And when you wake, you'll carry these mysteries with you. In every breath, every heartbeat, every thought, you'll be living proof that the most profound questions in science aren't abstract puzzles. They're the very essence of what it means to be alive. When you see a flower blooming, you might wonder how it knows when to flower and how it coordinates the development of its petals. When you watch a bird in flight, you might marvel at how its brain coordinates the complex movements needed for flight. When you look in the mirror, you might contemplate the remarkable fact that you're looking at yourself. These moments of wonder are not distractions from daily life. They're reminders of the extraordinary nature of ordinary existence. They connect us to the long history of human curiosity and to the ongoing quest to understand our place in the universe. They remind us that we're part of something much larger than ourselves, the vast interconnected web of life that spans the planet. The mysteries of biology also remind us of our responsibilities as conscious beings capable of understanding and protecting life. Our growing knowledge of biological systems brings with it the power to modify them, and with power comes responsibility. How we use our understanding of genetics, ecology, evolution, and physiology will shape the future of life on Earth. Climate change, biodiversity loss, emerging diseases, and biotechnology, all present challenges that require deep understanding of biological systems. The mysteries we've explored tonight aren't just fascinating puzzles. They're keys to addressing some of the most pressing challenges facing humanity and the planet. But even as we work to solve practical problems, the mysteries themselves retain their own intrinsic value. They remind us that the universe is far stranger and more wonderful than we might have imagined. They keep alive our sense of wonder and humility in the face of the unknown. They inspire new generations of scientists, artists, philosophers, and curious individuals to keep exploring and questioning. Thank you for joining me on this gentle exploration of biology's beautiful mysteries. If you found wonder in these questions, please consider subscribing to Sleep About Science, and perhaps leave a comment about which mystery intrigued you most. Tell us where you're listening from. Are you in a cozy bedroom in London, a dorm room in Tokyo, or perhaps under the stars somewhere in the Australian outback? Your stories and curiosity connect our global community of science lovers. And wherever you are in the world, whatever time it is as you listen to this, I hope these mysteries fill your dreams with curiosity and your rest with peace. May you wake tomorrow with a renewed sense of wonder at the extraordinary biological systems that make your existence possible. May you see the world with fresh eyes, appreciating the countless invisible processes that sustain life on our remarkable planet. Sweet dreams, fellow explorers. The mysteries will still be here when you wake, still waiting, still wonderful, still inviting us to look deeper into the magnificent puzzle of life itself. And in your dreams tonight, perhaps you'll find yourself swimming with bioluminescent plankton, flying with migrating birds, or diving deep into the molecular machinery that powers every living cell. For in the realm of dreams, we can explore the mysteries of biology not just with our minds but with our whole being, experiencing the wonder of life from the inside out. Rest well, and may your dreams be filled with the beauty and mystery of the living world.